Hi dear students, in this particular experiment or this task, I'm going to show you how to find the identity of a particular protein. After the sequencing of a particular protein, like once you do an experiment, like you might purify a particular protein and you will give it first protein sequencing and they will give you a sequence which is containing different amino acids. And you know each and every amino acid is being abbreviated here as with a separate letter. Now you have M here, it is methionine like that. Different amino acids have different abbreviated letters showing that which are these amino acids. So today we're going to find out using this sequence. You don't know what particular protein is this. You're going to find out using a bioinformatic tool what is the identity of this particular protein using a amino acid sequence analysis. So we are basically going to use a tool called the protein blast over here. This particular sequence is being going to be blasted against the database of various proteins which have been submitted in the protein database, data bank. So what we do is, if you have a particular sequence, amino acid sequence, you should copy that particular sequence. Now this particular region from M, you don't copy this one. This is the sequence identity, and this is the sequence has been explained here, sequence 1 and sequence 2, because you don't know what is it. You have to take from this towards the end, copy it from there, and then we will go and analyze. Now I'm going to analyze the sequence 1. What is that? So now I go to protein blast. I give a click onto protein blast and you just put the sequence over here. I'm pasting it over here. The sequence which I had previously shown you, I'm pasting it here. And you noticed one thing, this is a standard protein blast. If you're getting a nucleotide blast, you need to convert it into a protein blast. See, which is the option for that? You just look here. If you take blast N, it is a blast of the nucleotide. And if it is blast P, that means you are blasting a protein. And you go down, you don't know what is the particular protein, just give a blast option. There are different databases over here. I'm just giving you non-redundant protein sequences. There are different. You can either put it into a UniProt patented protein data bank. Like the different options are there. I'm just giving the default option of non-redundant protein sequences and just give a blast. And we'll wait for the results. You will get a window like this and it is searching, and the window will be automatically updated in 2 seconds. That's what they are saying. It's taking no seconds. 1 second. Okay, fine. Now you can see what is the information that you got. You get a sequence, the blast query, and the sequence which we analyze here is referred to as the query sequence. Okay, and what you do is, see, now it has been found to be as a translation elongation factor which has been present in Campylobacter. That is, they are showing 100% similarity to the translation elongation factor of Campylobacter, and it is having a query coverage of 70%. That is, among the sequence which I have given, 70% of that particular sequence is being found to be analyzed, and that is having 100% identity with the translation elongation factor. And this is the accession number of this particular sequence. So what you're doing is, your sequence of interest or your query sequence is being blasted against, or it is being aligned against various sequences which have been provided already into the database, and they show you the percentage of similarity. It is the same principle that we follow with nucleotides also, but here, since it's a protein sequence, that's what we are analyzing it against the protein databases. Now if I want to just, yeah, if I want to click onto this, I will get more details. Now when I press onto this, the query sequence, okay, what is the alignment with that? See, is there any, this is a query, this query is 72, as well as the subjects 34 to 93, that sequence has been found here. They are being found to be aligned because it's having 100% identity. You will have the similar one. Now, if you have dissimilarities between the two sequences, you will find gaps in between. We'll explore one with such an example also. Now I have pasted a sequence, another protein sequence, and I'm going to give it a blast. We'll see. We'll have a look what is that. It's just loading. 2 seconds. The database would also be a bit busy because simultaneously it can be used by different people from different side parts of the world. So it could be probably a bit busy. Now, in the second search which I had given, what else it means, I had deleted some of the sequences from in between. And you can see that here, after the results, it has been found that the query coverage is only 43% because I had, from the first file, I had deleted some of the sequences. So, the query coverage is only 43% here, but it is having 100% similarity with Campylobacter jejuni. And let's look a situation where only 98.65% is similar. Just have a look. How will it look? Now look at this. See, only here what happens? There is a 100% similarity. Here also there is 100% similarity. Yeah, they have loaded the ones which are having 100% similarity. That is, only identity is 74 out of the 75 bases are being analyzed here. The other ones are just getting loaded. We'll just wait for some second to see the other one. The alignments which are having less than 100% similarity. Now, if you go to look at this particular site, you can see that other than in the regions where there are 100% similarity, the total alignment between the two different sequences are happening. But coming to this case, you can see that in 96% similarity, there are regions where after M, here it is having T, and in the query sequence it is having T, and in the subject sequence it is having another amino acid called A. So the places where there is this thing, where there is a difference, there is a gap left between. So that's how you explore blast analysis and find out how much similarity of your particular protein is there with uh, a known sequence. Here also in 96% you can see there is a difference. A gap has been present over here. And hope so this is clear to you. Thank you for now.